I'd like to welcome everybody today to the Natural Areas Association's virtual symposium, White-tailed Deer Management in Natural Areas, Exploring New Approaches to an Old Problem. I'm Lisa Smith. I'm the Executive Director of the Natural Areas Association, and I'm joined by my colleague, Maura McGinty-Kloss, who is the Director of Programs and Communications. Maura will be managing technology behind the scenes today, and she'll be monitoring chat. So if you have any questions or you need any assistance, please feel free to reach out to her directly in chat or at info@naturalareas.org. I'd like to also offer special thanks to the Colcom Foundation from right here in Pittsburgh for supporting this program. Thanks to Colcom, we've been able to bring this program to you at a greatly reduced registration rate. So thank you, Colcom Foundation. And for anyone new to the Natural Areas Association, otherwise known as NAA, we are a national nonprofit communications and outreach organization that serves practitioners who manage ecologically significant landscapes to protect biodiversity. NAA understands that protecting these natural areas in perpetuity requires quality science to inform management practice and access to reliable resources. Given this, NAA curates science-based content for practitioners and researchers seeking applied science, evidence-based practice, and practical knowledge, and then solutions related to the management of ecologically significant lands and waters and the species that inhabit these special places. Our programs are designed to achieve on-the-ground outcomes and to encourage practitioner engagement and information sharing. There are many examples of our programming, which you can get familiar with on our website at naturalareas.org. One program in particular that I would like to highlight today is our biennial conference. The 2026 Natural Areas Conference, where science meets stewardship, will be held in Asheville, North Carolina next October, just about a year from now. Why do we say where science meets stewardship? Because the Natural Areas Conference is the place where land management practitioners bridge the gap between research and practice. The conference agenda is by practitioners, for practitioners, and it emphasizes applied science, evidence-based practice, and knowledge transfer to advance the day-to-day management of natural areas. The conference gathers around 350 natural areas practitioners in one location, and we feature plenary and concurrent sessions, symposia, roundtables, field workshops, hands-on workshops, and poster presentations. And there's lots of networking opportunities that we work into the week of the conference as well. So mark your calendars for October 5th through the 8th of 2026 and join us in Asheville, North Carolina. Be on the lookout for future communications that will include a call for proposals and registration information at the beginning of the year. And if you're not currently a member of the Natural Areas Association, I invite you to check us out. NAA is the perfect place to grow as a practitioner specializing in the ecological management of natural areas. And if that describes you, then you belong here. All right, a little housekeeping for today. Today's event is being hosted and recorded in Zoom webinar. If you'd like to access captions during today's program, please do so by clicking Captions on the Zoom navigation bar at the bottom of your screen. All registered participants will receive a link to the conference recording via email within a few days of the event. This link will be accessible for 14 days after receipt of the email. The only exception will be for those who purchase the team bundle that receive unlimited access. If you're interested in learning more about a team bundle after the program today, please email us at info@naturalareas.org or, again, put a post in the chat. For those of you who would like to follow the agenda during the event, you can use this QR code, which will take you to the detailed agenda on our website. You will be able to follow along on your phone. This is a 3-hour symposium that will include 3 presentation segments and 1 panel discussion. There will be a 10-minute break at approximately 1:30 p.m. Eastern Time. Each session will include a brief opportunity for questions after the presentation. And speaking of questions and answers, we will try to pose as many questions as time permits before moving on to the next presentation. Please post all questions in the Q&A located on the navigation bar, again, at the bottom of your screen. The Q&A function allows the hosts and presenters to better manage questions versus trying to keep track of them in chat. Please note that there may be a few questions that will be held so that they can be addressed during the panel discussion at the end of our symposium. We encourage everyone to participate in this symposium using chat to express ideas, make comments, or to share resources. We know you want to hear from today's presenters, however, they also want to hear from you. So please engage with one another as we work together to identify new approaches to the very old problem of white-tailed deer impacts on natural areas. Why are we here today? White-tailed deer are one of the most persistent and complex challenges we face in natural areas stewardship. Overabundant deer reduce biodiversity, we know this. They prevent forest regeneration, and they impact ecosystems in profound ways. We're learning a lot more about these ways as time marches on, with deer populations being left unchecked in many places. The management of white-tailed deer is not new. However, the growing challenge of overbrowsing is elevating the need for information sharing and creative solutions. Today's virtual symposium is bringing together leading scientists and practitioners who are focused on this issue to provide perspectives on where we as a society sit with this issue in 2025, to share recent evidence-based research on deer impacts and new insights from a 30-year exclosure study, to highlight a scalable approach being piloted in Virginia that brings together wildlife managers, natural heritage programs, and private partners, and to discuss what we are learning both from the presenters and each other to address this ongoing problem. Before we get started, I'd like to introduce NAA's partner in developing this program and today's panel facilitator, Dr. Ryan Klopf. Ryan is the Mountain Region Supervisor and Natural Area Science Manager for the Virginia Natural Heritage Program within the Virginia Department of Conservation and Recreation. Ryan is a longstanding supporter of the Natural Areas Association as a former board member and current chair of our Science Advisory Committee. Ryan effectively works in both the science and practice realm of natural areas management, as well as effectively representing the perspective of the land management community on the panel. In order to be efficient with our time, I'm going to introduce our first two presenters, and then we'll have a break, and I'll introduce our second set of presenters. Our first presenter today is Bernd Blasi. Bernd is a professor directing the Ecology and Management of the Invasive Plants Program in the Department of Natural Resources and the Environment at Cornell University. Bernd develops and implements biological weed control programs with an ever-increasing focus on the impacts of multiple stressors, including invasive and native plants, earthworms, slugs, and deer on a wide range of native organisms. Through involvement with deer management at Cornell and surrounding municipalities, he has created a network of exclosures to study impacts of deer on many species and processes. In addition, he is developing bioindicators to assess effects of different stressors, including deer. He has also commenced a large study in northern Wisconsin to assess the effects of wolves on deer and forest plants, a trophic cascade. The ultimate aim of Bernd's work is to increase the conservation values of all lands through development of best management practices. Our second presenter is Dr. Susan Kalis. Susan received her Bachelor of Science degree in botany from the University of Michigan and her master's and PhD degrees in ecology and evolutionary biology from the University of Chicago. Her research focuses on the evolution and ecology of plants, including stressors that influence native plant species population dynamics and the effects of overabundant deer and exotic invasion on the composition of understory plant communities. Over her career, Susan was a faculty member at Michigan State University, the University of Pittsburgh, and the University of Tennessee, Knoxville, where she served as professor and department head of ecology and evolutionary biology from 2015 to 2022. She is currently a research professor at UTK and continues her long-term research at Trillium Trail in Pittsburgh, Pennsylvania. And now I'd like to invite Bernd to turn on his video camera. Welcome, Bernd. We look forward to hearing from you today.
So thanks for the opportunity to address an audience, and if I see it from what was in the chat. It's Midwestern and Eastern based, as is probably pretty typical. I'm charged here with addressing part of what we know about the deer issue. Lisa already presented or mentioned a few things. Um, but our knowledge base, I would say, is still extremely limited. We don't know really what's happening locally. Do we really know everything about the impacts that deer have? Are they really endangering fauna and flora? How do we know it's deer and not global climate change or pollution or something else? And can we do something about it? I'm not going to address much on that one, although we have done a whole bunch of work trying to enhance our ability to manage deer. And I will get a very brief mention of that. But I wanted to preface something here because that very often comes into contentious debates about deer management, lethal management in particular. I care and I want to protect native species and ecosystems. I come to the realization that there is no conservation possibilities, at least in the eastern landscapes where I work, without greatly reducing deer abundance. So I manage deer to prevent negative ecological and a little bit economic impacts, but I'm driven by their ecological impacts. So my central claim, and I will explore some of that, I'm not going to go through entirety of this, is that deer impacts um, on ecological and human health are vastly underestimated. So deer can create pictures like this, can have influence on other invasive species, on tick abundance. They're really at the center of an ecological and economic crisis. They affect pollinators and birds and insects and salamanders and all kinds of things. So my central claim is we know some, but not enough. And as we continue to look into issues created by deer, and it's not their abundance, it's their impact, we learn more and more about their central role in structuring ecosystems. Um, do I really need to talk about this? Well, we have lots of evidence, lots of papers. Some of them are from my group, some of them from many other places. There are reviews out there. You can look at that. And what you will find is that it's most well known for plants and forest regeneration. Lisa mentioned that already. Here are some of the papers. There are hundreds, maybe thousands out there that will summarize it for the US, for North America. And we are not alone in that because the same problems plague Europe, and many other places where deer are introduced or native. Um, what we can say, it's not only affecting all the landscape, but it's also affecting our cherished natural resources, national parks, natural areas, whatever you have. Here, two examples from Catherine Miller's work, really using FIA data, forest inventory analysis. And we're looking at the major regeneration depths across the entirety of the Northeast and Mid-Atlantic states. That's where she focused her work, and it's affecting the national parks. And I'm not going to go into a lot of details, but I believe these things are freely available. But even in national parks, along that gradient from really Virginia up to Maine, national parks are in a slow motion collapse, meaning the forest, these are mostly forest systems. The overstory is still there, but the recruitment is not there. So they're in a slow motion collapse. As individual trees die, they're selectively replaced by those that can withstand some deer browse pressure, but overall the systems as we know them will be changing dramatically if we don't do something about it. And how do we know it's deer? Sometimes you can just watch. This is a single doe in the village of Lansing on Lynn Leopold's place. We planted oaks out there, and it doesn't take a lot of deer to decimate the oaks. You can kind of see that's almost all that there is on the forest understory. And oops, somebody may be watching. So it doesn't take many deer to create a major impact. But it's difficult to demonstrate that the deer actually have an impact because here's an example on the right. There's some browsed ginseng. Okay, you can find that. But the absence of individuals is sometimes very difficult to indicate. So we have here a trillium meadow, wonderful trillium expanse. In the lower part of the picture here, every single flag is a browse trillium stem. and they will selectively browse the flowering individuals. The absence of these flowering ones is really difficult to document until you go in. So I had to put flags in there and say, this was probably a single deer going through there and eating all of those. And I know Susan will address some of those things that she has done there as well. So this is a major impact, but you may not be able to detect it if you were to just walk by and don't do a critical assessment of what was happening there. There was a Pennsylvania deer experiment where they did a wonderful thing of trying to do different deer exclosures with different deer density and then see what regeneration would be happening. They had to abandon this, I believe, after like 10 years because it created a problem for the high deer abundance enclosure. They were not exclosures because deer health was going down. And what Again, you can find this in Ecosphere by Nuttall. There's other papers coming out there. Basically, deer create a domino effect of reducing diversity, abundance of primary producers. That goes in and affects insects and their consumers, which here is just depicted as birds. So we have an entire rippling effect that deer create at different densities. And the higher the deer density is, that's what you have here on the y-axis, the more detrimental their impact is. Here it's measured as deer per square kilometer. Very often you have deer density in suburbia that way exceed what they were able to accomplish here in their deer enclosures. So we are creating unhealthy landscapes. That's depicted here this is mostly from New York. We now also recognize that deer can almost be like moose going in after aquatic vegetation. This is some pickerel weed that should be very abundant in my ponds here, but the deer go in and almost feed like moose, including sticking their heads underwater or into the water, not underwater. And that creates trickle-up effects or trickle-down effects, right? If there's no vegetation, there's no insects that can live on them. We know about pollinator declines. This is a major issue for many landscapes, not just in the Northeast or the Midwest. Dale Schweitzer did an interesting assessment, and he looked at Macrolepidoptera in New Jersey, and he found that in the forest canopies that are still existing, including in the pictures that you see here, most of the known Macrolepidoptera were still existing. In the forest understory, the ones that are specialized on herbaceous ones, most of them could not be found anymore. So he contributed to deer browse. So we see a lack of understory, lack of the shrub layer, and the transition from seedlings to adult trees is not enabled. That affects entire components of the ecosystem, not only from the primary producers, but the consumers, litter levels, and other things. So we have evidence there. And I engaged in that starting in the late 1990s with a big work at West Point, trying to understand what we call multiple stressors. Here, putting deer into a picture together with introduced earthworms and introduced plants, slugs, and some climate change. Did it with a lot of people that contributed to all of this. Can't mention them, but you will see their names pop up in various publications that they're out there. And by the way, most of these publications that I will reference, at least when they come from my lab, are freely available. So we use something, a change. We do work with fences, but we use an approach that sometimes make people scratch their heads. We use mark recapture using plants. Well, the plants are always there, are they? Well, I will show you that they're not always there. We also don't always find them. And deer have sorted out plant communities. So to really understand what their effects are, we may need to bring in individuals or certain species. So we're actively transplanting individuals. We mark them individually in open and fenced areas. We try to create experiments that work with and without earthworm invasion, with and without native plants. We call that multiple stressors because they all have an influence in what deer effects are and how deer use landscapes. We record survival, growth, and reproduction, which largely will go into demography. I know Susan will talk about it, but we're focused on that one as well. We're trying to be very long-term because effects are not very quick. Forests change incredibly slow. And we use many different field sites because in our investigation, site is always significant. That could be previous land use history. It could be all kinds of things, different fertilities. Many different factors would influence how sites behave and respond to what we do. We measure browse directly. So to do that and be able to compare across sites, we developed new assessment protocols. I'm going to go into that one in a little moment. And we assess other factors. So on the individuals that we plan, we can detect whether it was a deer or whether it was an insect, or I forgot even rodents to put in here. But disease and competing plants all will have an effect on how well our individuals that we typically call sentinels will actually perform in these systems. And instead of just focusing on primary producers, we're incorporating other biota, amphibians, salamanders, birds, and vertebrates into our assessments. I will cover some of these, not all of them. So just a little picture guide here. What do we do? Hey, our plants are being marked individually as seedlings. We go out, we measure them, we give them numbers, we tell them, hey, where are you? How are you doing? Are you flowering? Are you surviving? Those type of deals. We also do typically other things, vegetation assessment following individual seedlings that we mark with bird bands. So we go in these dense vegetations. If you mark these seedlings, you need to find them again. So if we mark them individually, we can use a metal detector to record them again. We do transects, just a little picture guide of what it may look like when you walk into Blossy Lab. We started out with an exclosure network at 12 different sites in the West Point area. We then expanded it towards Cornell. So the big 3 circles there are different exclosure systems that we have around Cornell. We increased the size because we realized that edge effects are huge. And we now work in our largest exclosures. There are 2 hectares, 5 acres each. Well, I need to recognize to all of you or admit to all of you, it's a deer reduction, not a complete deer exclusion, because trees always fall on our fences. They need to be repaired, and we can always find some deer browse on the inside. So, but we're trying to maintain them. One of the things that we realized working with exclosures is doing the typical plant community assessment is basically useless because these forests change very, very slowly, and the plant community composition is incredibly slow to change. So you have here, we have vegetation cover, species richness, the height of the vegetation or litter volume. This is like what is it? Like a four-year assessment where we did this, and we realized we can't do that. We need to find something different because the plant communities have been sorted out by deer. Only the ones that can exist in high deer browse intensity are still there. The other ones need to be reintroduced, and they're not just coming. There's no seed bank of the charismatic understory flora that we typically have. So we need to assess things different. Nevertheless, what we found with our exclosure studies is deer exclusion reduces non-native plant abundance. Here, I'm just focusing on 2, which is garlic mustard and Japanese stilt grass. Inside the fences with deer exclusion, very little garlic mustard, maybe even going almost completely away. Same with microstegium. So we can manage a number of invasive plant species by excluding deer. We also realized we can manage introduced earthworms by excluding deer. So this is a long-term study that Elizabeth Simpson did in Pennsylvania and New York. We were the first to document this really, as the longer you have these deer exclosures out there, the more the earthworm abundance goes down. That has now been repeated in Indiana and Ohio and Wisconsin. So that's a persistent change. And sometimes it almost goes to complete deer exclusion. Fencing effects become stronger with fence age. So what we have, I need to go back to this one. What we now say, we're creating a fence effect. We are not creating a deer exclusion effect because fences do more than just exclude deer. Litter will accumulate, earthworms will go down, all kinds of other things are happening in response to deer exclusion. So we call it a fence effect. Then we try to combine the effect of deer with earthworms in forests that are invaded by earthworms or not invaded using fence exclosures. And this is work by Annis Dobson here. And we again planted out seedlings of 20 different species with and without earthworm. And you can kind of look along here and say, oh, for many species, survival from the little seedling stage to existing plants is very low for most of our forest understory species. It is affected whether things are fenced on the left side of each panel column here, or whether they're not fenced. But for many species, it's not. Actaea rubra doesn't care whether you're fenced or unfenced, or whether you have earthworms or not. Other species have incredibly high survival rates, like Geranium maculatum. Again, they don't care whether you have earthworms or not, or whether you're fenced or not. Other species respond strongly to earthworms or not. Here, Caulophyllum will be reduced in terms of survival where you have no earthworms. So they benefit from earthworms. So which species you pick, how you assess them, how long you run your things will actually affect what the outcome of your experiment may show. So we need to be careful in how we're doing this. Sometimes things change over time, things reverse. For Polygonatum virginianum here, you can kind of see that what we early had over a longer time, the effect of earthworms will shift. The ones that can survive high earthworm numbers may benefit as adults being more reproductively active. So what does it do when you fence things? You all may know, hey, reproduction goes up, plants start flowering, become bigger, and that has obviously demographic effects for the long-term survival of populations. Here's just an example from false Solomon seal. It's orders of magnitude more number of seeds that are being produced and potentially result in plants on the inside. Here they call protected versus unprotected ones. Take some time to do it. Susan may talk more about that. But we're also finding things like it affects not only the number of seeds per plant here on the left side, but also whether plants actually show up above ground. By having individually marked plants, we can go back and say, are you still here? Have you survived? And we discovered that white trillium and also red trillium take years off, like orchids do. 1, 2, 3 years we have now documented where they do not show up above ground. So going out and counting a number of flowering trilliums or existing trilliums as an assessment of deer browse intensity will not work. Those things fluctuate from year to year, but you can kind of see the proportion of dormancy is very low when they're protected behind fences and goes to 20% in the open. Oops. My picture is gone. So I need to go to something. So we worked a lot with fences. We were not trying to downplay the importance of fences, but we also know that having fences in the landscape is not something that we can do everywhere, and it doesn't really respond to the reality that most of us are facing. We can't fence all the places. What was supposed to be here on the left side was a map of Wisconsin showing the return of wolves from Minnesota into Wisconsin over time and wolf abundance. We used this area and had 40 different sites that were structured across this gradient of wolf residence time and wolf abundance in northern Wisconsin to try and then understand, are they helping us? Are they reducing deer browse? Are they doing something to the vegetation? And how are birds being affected by it? I thought I would have this paper up for you. You can see this is only an author proof. It's an ecology and evolution. It should be out every moment. This is what you need to look for. I don't know whether the DOI will be the same. It was not live as of this morning. So we did this, as I said, in sites that were basically the same mesic forest type that we used. We looked at, we planted out sentinels in these areas, and we used red and white oak and zigzag goldenrod. So when you see here on the right side, the proportion of sentinels browsed, that's the frequency, or that's the proportion of the individuals that were browsed along these different sites, 40 different field sites. Remember, that's what it is. And we recorded the abundance or frequency of 11 common plant species that were common in these 40 sites. And what you can kind of see is that these plants, the presence of these different plant species, this is not the same order, goes down as the proportion of sentinels, the ones that we planted out purposefully, increases. That's a trend for almost all of the species, regardless of whether there are being deer candy or not. So deer affecting more than just the ones that they are eating. And we see a strong effect on the plant populations. That will take time to really sort out, but we have a strong effect of deer browse intensity on associated vegetation. Oh, come on. Let me tell you what this slide shows. I don't know, in the pre-screen it was all up. What we see here is how am I going to deal with that? What we see here is a graph of the abundance of singing males at these 40 field sites as a function of deer browse intensity on our sentinels. So basically a similar thing as you see here, proportion sentinels browsed on the x-axis, and the abundance of singing males is the number of days that they were singing. We used acoustic recorders in the woods that were there for 2 months, recording every incidence of singing males, and then we analyzed that, and we focused on understory birds. The graph here was just showing 4 different ones, with ovenbirds being flat, veery going down, hermit thrush going down further, and song thrush going down even further, as in response to increasing deer browse intensity. So the only reason that ovenbirds didn't respond to that is they make an oven, as you can see on the right-hand side. All the other ones are nesting low on the ground directly, and have open cup nests. So if there's no vegetation or reduced vegetation, these nests will either fail, or the adults will abandon them, or not even select those sites. So some of the most common birds that you will find will be negatively affected by deer browse intensity. I apologize that this picture doesn't show up. So what's really important for us is to understand how deer affect the different individual life stages of birds, of trees, or of herbaceous plants. We know that deer will not directly affect an adult bluebird. It will indirectly affect maybe food availability. It will not go into a nest, but basically the population dynamics of bluebirds or of oaks or of red trilliums are affected how well they transition from juveniles into adult ones. And it's the same. You can make demographic models. Susan may talk a little bit more about that. But if for some reason the transition from one stage to the other is not okay, very few survive from seedling stage for the oak, for example, into a sapling stage. There's a problem if that's due to deer herbivory. So we need to understand how deer affect the transitions in the life history matrix of these different organisms. That's what we're after. And if you don't do that, you can come to greatly different conclusions about the health of ecosystems in response to deer. So I need to go here. We had a deer summit. David Steinbrook, who's with the Pennsylvania Game Commission, said deer impacts across Pennsylvania are fine. They're at acceptable levels or even good forest regeneration. Deer impacts are only high, the red ones, above acceptable levels in the northeast corner of Pennsylvania. This is forest inventory analysis, number of seedlings. Compare that to the analysis that Catherine Miller and Brian McGill did, where basically the forest regenerations and the number of seedlings per square mile is dismal in Pennsylvania. Same data set, but The Pennsylvania Game Commission focused on seedlings. Catherine Miller and Brian McGill looked at the transition from seedlings to saplings to trees, coming to an extremely different conclusion. And so the Pennsylvania Game Commission basically cheats themselves out of their responsibilities to manage deer differently by going to seedlings. So we wanted to understand, how can we get away from pre-existing conditions basically that you have in the landscape where you go out and trying to assess that. So we developed a new study method development, we call that. We used that then in the Wisconsin study that I referred to, and we developed indicator. We focused on red oaks initially. Then Brendan Quirian expanded that to including goldenrods and asters to trying to have more sentinel species available for us and trying to understand how that would influence the browse frequency. You can read up about that. They're publicly available. We tried to understand what Cornell deer management would do to the browse frequency. We needed to do that because there is no ability to compare across sites, but using, because the plant communities may be different. But we use this to trying to understand how increase in hunting pressure may be affecting the browse rates, right? So from left to right, you would see an increase in the deer browse in deer management from nothing in Cayuga Heights at some point, little community outside Cornell, to Cornell areas, Finger Lakes Land Trust, state parks. This is all in Tompkins County or surrounding Cornell. This is Atkinson Center. These are 2-hectare exclosures. This is the property that I live on here and sit on right now. And we compared the browse levels here to what was happening to the white trillium to have a comparison. So as hunting and lethal management pressure increased, we can drop the browse rates to below 15%, which is kind of like a ballpark situation. You need to have the annual browse rate below 15% to allow an oak to actually grow and become a sapling and a young tree. With heavy management, I'm able to reduce the browse rates on oak. So my oaks here are recruiting again. I can have oaks unprotected and they're growing. Can't do the same with trilliums. So the browse level, oh, come on. So the browse level that I have here is not acceptable at this time. We need to reduce the deer even further. Because there are no pictures there, I'm just showing you different forest communities, and it's a problem that we can't standardize from one community to the other to measure the same thing. So our sentinels, our planted individuals, allow us to then compare from one side to the other, even though the land use history may be different, tree communities may be different. With the data that we collected, we were able to show also that oak browse and the frequency of oak browse is a good indication for the number of deer in a certain area. We took advantage, and most of you, maybe all of you, will not have the advantage of a marked population of deer that run around, so you can assess their abundance. So over time, with lethal management, shooting at night over bait, with crossbows and bows around Cornell, we were able to drop the deer abundance down to half of what it was previously. The way that we did it, allowed the browse rates from 60 to 80% go down to 25%. So we did good, but not sufficient. At these browse rates, we still don't have any oak recruitment on Cornell grounds, but because we were able to do this, we were able to continue the justification, continue deer management. As I said, it's not sufficient. We need to do more. And recreational hunting was not able to help us at all. So what I'm trying to say here, we developed indicators that allowed us to assess deer browse intensity on plants. I'm not showing you the ones on salamanders, or I showed you the ones on birds, where you measure something directly. Even though it's also a legacy effect. We measure, not infer, deer browse intensity directly by our sentinels. These sentinels are then sensitive to changes in deer browse pressure. For example, if you change hunting or management activity. We have with this, because we replant every year, a rapid response assessment capability. So if you change your management of a deer, you want to know now, not in 10 years, whether you were successful, because that's the only way that it's socially acceptable. As I said, we have this method that then allows a standardization among many different sites with many different plant communities, many different years, or even regions. Now, what we have with red oaks and the asters and solidagos will not work everywhere, but the approach of using sentinels and planting them out will work everywhere once you select the right species. They can capture impacts or stand in for other species. Think about the browse intensity versus birds, or browse intensity versus occurrence of other plant species. By planting out, we have the ability to incorporate legacy effect, but are not enslaved to legacy effects, because previous agriculture or others may affect the way that plants grow. It's appropriately scaled. We can do it for individual landowners or for forest sections, and it allows the participation by citizen scientists. So we not only do the biological stuff, we also try to understand, can we, with what we're doing, gain public support for deer management? Because all of you will be familiar with the controversies that are erupting once the change in suburbia or somewhere else goes, and you want to increase lethal deer removal. The decision makers and agencies typically hear amplified and very well coordinated messages from two particular groups. One is hunters, and the other ones, I basically group them as animal welfare groups, whether it's the Humane Society or PETA or whatever else you have. Those have amplified voices, very well coordinated. They are loud in social medias and everywhere. But are they actually representing the attitude of everybody? Management agency also typically sample or trying to sample attitudes of their residents or citizens, but very often they're not using very sophisticated questionnaires. Sometimes they only sample hunters, or then they have very few options. Do you want more deer, keep populations the same, or do you want fewer deer? And most people will then defer to the agency side. You are the ones that have the expertise, you tell me. So very often it means keep populations the same. That's the outcome of very simplified questionnaires. But is lethal deer reduction finding public support? I'm going to go to the next slide and explain you how we did this. You can find this study openly available. This is for New York, but we were asking people whether they would accept lethal deer management if that would reduce to reductions in Lyme disease or road safety or reduction in starvation of deer, improving in forest health, help climate change, wild plants, and so forth. And you can see the overwhelming majority of people will accept lethal deer management for these particular reasons: economic, human well-being, and ecological well-being. Please note that wild plants here, protection of wild plants is much more important than the protection of garden plants. So people were looking at this very critically in the way that they were answering these. There's 1,500 people, according to the makeup of New York residents that we asked. We see strong support for lethal management, providing hunting opportunities, very little, well, much less support. Only 50% of people would support that. But the support is linked to the goal and the accomplishment, so you need to measure. Our Sentinel protocol can deliver that. It's part of accountability. We have done very similar surveys in Scotland, basically the same results. We have expanded this to capture states, not just New York, but the Northeast, Midwest, Mountain West, so Idaho, Utah, Wyoming, and Pacific Northwest, and the results are basically identical. So it doesn't matter what the political leaning of individual states or regions are. Once you link the outcome, the ecological outcome of lethal deer management to these goals, the support is very widespread. There's also no difference between rural and urban residents. They agree, so that there is, that people become weakly associated with lethal management once they're urbanized. It's just a myth. So, as I said, very similar support or very similar work shows that support out there. So if you decide that you want to protect your local resources, and the cherished importance of natural areas, you will find public support as long as you justify it in the right way. So ultimately, the decision is ours, whether we want the pauperate situations with lots of deer, or whether we want mixed vegetation with lots of abundance of birds and plants and the cherished ones that we have, plus some deer. I think we can make that decision. We need to have new tools that we need to develop, but it's possible, and we will gain public support for that. Thank you very much. That's what my presentation is about.
Thank you so very much, Baron. That was fantastic. We do have a couple questions for you, if you've got the energy for it.
Yeah.
Okay.
I'm gonna stop sharing here.
Okay, great. The first one is what would be the mechanism that reduces earthworms in the exclosures?
I've been pondering that for 10 years. Others have done that too. We don't know the mechanism. We may think it may be microbial. So some linkage, but we really don't understand what it is, because obviously the deer are not, breeding earthworms or doing anything. We don't fully understand that yet. I'm not a microbial ecologist in that sense. The pattern is repeatedly being found, so we're not the only ones documenting it. That's why I'm saying I don't have the mechanism. I'm sorry.
Okay, we'll figure that out. Next question. We can't fence everywhere, but should preserves fence at least some of their property to establish refuges where plants can be protected until browse is reduced in the future?
I would agree with that. So what you want to do and where you want to focus your management efforts, and we will hear more about that, what Virginia is trying to do, right? Or is doing already. The high quality ones, you need to protect those. That's the future of the forest. That's the future of the understory, right? If you have valuable plant communities, vulnerable or whatever it is, if you can fence them, please do so, because that's the inoculum for other things to grow, for places to spread. It will take decades, unfortunately, right? Our forest understory communities are incredibly slow. They're made to live long, have low seed output, and for me, they're beautiful, but they need some protection, and we can't wait for all the deer numbers to go down that they can thrive. If you can fence, please do so. Larger is better.
Very good. Okay, and then back to, you defined this early on, but once again, the definition of sentinel.
Yeah. So sentinel is basically any species that gives you a signal of the status of the ecosystem, right? So think about the canary in the coal mine. That's basically the thing. It's an indicator to the health. When you go and visit a doctor, your temperature and heart rate is being taken, or your weight or whatever. So it's an early, or is a signal that stands for something different. So we validated our oak protocols. Well, hey, by measuring the browse rate, we get an information about the deer abundance in a particular area. I don't necessarily care about deer abundance. If they wouldn't do anything as standing around and being the target of photographers, but they're doing that, they're having an impact. So sentinel for me, there's literature out there where you can do more, but it's an indicator that it transfers information about the health of the system. Here, about deer browse intensity. That may not be quite as good as the. So I'm happy to follow up on that.
Okay, great. And I'm going to leave a couple of these for you to, answer after you're done.
Don't have any high hopes. I see the one about sterilization. No, absolutely not. No impact, nothing, zilch, zero.
Very good. Thank you for that. Okay, let's see. Maybe one more. Could Bern touch more on the effectiveness of plant community assessment in terms of deer browse, abundance, activity levels? Would a study examining the difference in plant diversity at various locations in an island archipelago in relation to deer activity be suitable for a master's student, or would the study be not useful?
So the question is, what archipelago are deer there? Are they colonizing? Do you have reference sites? There are particulars. But what we know, when you had deer in high abundance, they have sorted out the communities. So you putting up a fence around an area. I mean, Susan will talk about a beautiful place, right, that you could protect that was still very diverse and could respond. We see our sites that are high quality, they can respond to protection. The ones that are a little more deteriorated, they have lost their ability and to species composition to respond to deer exclusion. So the particulars, I'm happy to follow up with whoever asked that question. But we know with the work that we did, measuring community responses will not work. But for a master student, absolutely not. No change from one year to the other. That may be annual fluctuations. You may need to go and look for decades, right? So when the foresters talk about a century of change, that's kind of what plant communities do. I couldn't show some of the slides that I have showing how our communities change with fencing over a decade. Susan may show some of that, but it's incredibly slow. So you will not pick up a signal. So I would discourage that just on the face of what I'm hearing right now.
Okay, very good, Baron. Thank you so very much. If there are any questions that you might be able to answer, we may pull.
I'll go into the chat and see what I can do,
Okay.
and I'll try that, or the question and answers. Yeah.
Very good, very good. And
Thank
with
you,
that,
everybody.
thank you very much. Oh, there is one question real quick about your presentation and
Yeah.
sharing a presentation where a couple of those slides were.
So most of the ones that I showed,
Yeah.
with some of the exceptions about the birds in Wisconsin, are available free without being behind a paywall. I'm more than happy to provide a list of references to you and said, you can find this all here.
Okay.
So I'm hesitant to share that one, but my slide will be available. Okay.
All right. Thank you very much. And with that, we'll go ahead and kick the program to Susan. Thank you, Susan.
All right, so I want to thank everyone for attending and for my opportunity to participate in this symposium. I think that my title, Learning from a Long-Term Exclosure Experiment, kind of says it all. I'm trying to present to you what we have learned since the original talk that was given at the NAA meeting in 2009. So my work has been at the Trillium Trail Reserve, which is going to be abbreviated TTR, which is in Fox Chapel Borough near Pittsburgh, Pennsylvania. In 1989, this is a photo that was taken, the one on the left, showing just hillsides covered with trillium, the thing that we all wish happened everywhere. And just a few years later, in 1992, people went out to see the show, and basically there were barren hillsides. This prompted Lisa Smith and Joanne Davis to set up some deer exclosure studies. And when I moved to Pittsburgh in 1995, I started working with Lisa and my graduate student at the time, Tiffany Knight, and Jessica Dunn, who was in my lab as well. With the funds from a lot of different people, started collecting data in the area. First of all, Fox Chapel is a very wealthy community, and when they saw what had happened at Trillium Trail, they paid for aerial flyovers, and you can see that they did this starting in 1993 and continued into the early 2000s, and the historical deer abundance is somewhere around 10 to 12 deer per square kilometer, and the numbers they were measuring were 4 times that. It was really pretty stunning. And we started collecting data right away, and we're seeing shifts in percent cover and other things. And based on our early studies, the Fox Chapel Borough decided that they would fence, as Bern was just talking about, the core of the wildflower reserve and they hired sharpshooters to control the herd. So they were doing things proactively to try and protect this beautiful area. So in 2009, we demonstrated a link between deer forest invasion and understory community change. So if you look at this figure, you can see that where deer are excluded, we have a majority of white trillium and lots of other species present, but where deer have access, we have this spike in the relative cover of garlic mustard and then some other species that deer don't like to eat. We also saw an increase in the percent bare ground with deer access, whereas where deer were excluded, the plants recovered, the community recovered. And so we had this dramatic thing that we could present at the natural areas meeting about how deer are linking the invasion of forests. And this is what it looked like at the time. Here's the deer excluded. I mean, you can just look at it and see it's pretty dramatic. So these kinds of things have been shown quite often now. People are very curious about, showing this is a very repeatable thing. But what we were really interested in is understanding what are the potential mechanisms that are driving these early findings at Trillium Trail. We're interested in whether the native palatable species were showing demographic shifts, whether the forest understory as a whole was shifting, is our invasive plants in general succeeding? How does it affect the soil and root fungal community shifts, abiotic changes to forests, and in addition, unpalatable native species success? Are they going to be doing a lot better because the palatable species are declining? So we built exclosures that were large, 14 meters on a side, and there was a paired fenced, fenced and unfenced. We gridded the area inside of each of these plots and tagged tens of thousands of individual plants to understand their demographic transitions. That was what Barron mentioned. And in each of these paired plots, we allowed Alaria to invade, or we weeded it on an annual basis. This is what it looks like with some captive undergraduates inside the fence helping to collect the data. There were lots and lots of people who have worked on this study since the beginning. It's been a huge amount of work, and I am grateful to everyone here, but I'm not going to mention all their names, and also to the National Science Foundation that has funded this project for more than 20 years. I also had squads of undergraduates work with me between 2026 and 2025, and the project continues. So what we were really interested in is understanding how plants interact, how the forest invader affects these interactions, how the forest invader affects the AM fungal community, how deer affects soil and herbivory, how these things feed back to each other, how the light environment is changing. In addition to our plots, we were interested in seeing if this was generally applicable. So we had study areas beyond the Trillium Trail, exclosures and control plots. We had plots in northwestern Pennsylvania and also in Virginia and Illinois. The focal native perennials were jack in the pulpit. Deer don't like to eat this species. Smooth Solomon seal, fall Solomon seal, and then white trillium and wake robin. These two are indistinguishable when they are non-flowering. So they're sometimes grouped into the trillium bin. So just so you know, these species are extremely long-lived. Trillium can live more than 100 years. They grow a rhizome and these very interesting rubbery roots that are packed with AM fungi, and they grow slowly. So what I'd like to do now is go through some of these circles and tell you what we have found about the mechanistic effects. So we know from our early work that the annual percent of the focal palatable species, these are individually tagged plants that are browsed by deer, and we looked at them every year multiple times over the growing season, that the average browse rate is high, it's 22%. And what we found is that when we exclude deer, the fraction of plants that are able to recover from the loss of carbon that browsing imposes on them and start producing flowers again goes up, but it takes time. And by over 5 years, basically, we saw a dramatic increase in the fraction of plants that were flowering inside the exclusion, whereas only a small fraction of the plants in the deer access plots were flowering, and it really didn't change. So these demographic shifts that Baron mentioned, and I'm really interested in, require very specific kinds of information. So what you have to do is measure the transitions across each of the life stages and follow them over long periods of time to understand where the plants are most sensitive to the effects of deer browse and how it sort of breaks up the demographic matrix projection. So what we see is that when plants are not eaten, they maintain their reproductive status. And in fact, if they're not eaten for 2 years, they can maintain high rates of reproductive status. In contrast, those that are eaten for 1 year, basically it's cut more than in half, and most of them regress to a non-flowering 3-leaf stage, and about 8% of them become dormant. And are you seeing this? I hope you're not seeing that. You are? Are you seeing the little, somehow I'm getting this like chat box and stuff showing up, but hopefully you're not seeing that.
No.
We can
If
see
they're
your
eaten
slide.
pardon me?
We can see your slide.
Okay. If you're eaten for 2 years in a row, then the probability of being dormant or being reproductive goes way down to only 19%. The majority of the plants regress to being 3-leaved. So when you look out in the field of Trillium, it doesn't have any flowering individuals. They could be there as above ground 3-leaved individuals or dormant individuals, but it's very, it's extreme what happens to them. And so in the 12 sites across Western Pennsylvania, where we use these demographic transitions that were measured on individually tagged plants at different stages, we found that the higher the average deer herbivory, the lower the population growth rate. So a population growth rate, lambda of 1, means that the population is stable. And so what we see is if the browse rate is less than 15%, these populations are stable. But if it's higher than 15%, you can see that the lambda, the population growth rate, is sinking. And so these really high levels of deer browse create unstable populations, that is, populations that will decline over time if the level of herbivory continues. So what's some practical information for managers? If the number of native species that are flowering is very low, and you've seen a drastic change, deer use of the area is probably too high. And if this low number of individuals is maintained over time, there's a problem. And if average browse on native species exists roughly 15%, this is what we found for Trillium, but it's probably works for other species, then those populations will decline. And your native populations are in trouble. So what I'd like to move next to is some demographic work with invasive species. And what we did at the Trillium Trail Reserve, the whole experiment was set up to be analyzed as a life table response experiment. So you can compare the treatment effects, in this case, deer versus no deer, and how they affect garlic mustard or Trillium over time. And I'm not going to give you any of the specifics of the methods. I'm going to give you the big picture. I can answer questions about that afterward, or I can give you the reference. So what we do is have these matrices for garlic mustard and Trillium, and we use them to compare the contribution of each of those transitions to the population growth rate. And what we found is that for Trillium, that there was a 16% increase in the population growth rate when we excluded deer, and that's huge. That's a huge effect. And what we see is that we've lost, if you look here, the dark bars are the flowering individuals, and you can see there are very few flowering. These are numbers of Trillium individuals. And in addition, we've recovered the recruitment of seedlings after 5 years where deer are excluded, and very few seedlings in the deer axis. When we compare what happens between the Trillium and the garlic mustard, we see that the exclusion of deer has a positive effect on every transition in the Trillium's life cycle. And the most important is the remaining flowering, because those are the individuals that are dumping seeds into the soil that can create the next round of recruits into the population. Conversely, for garlic mustard, every contribution is negative in the presence of deer. So the difference between deer access and deer exclusion makes garlic mustard tank. And so if we look at the population growth rate of garlic mustard in the deer axis, we have no difference. It has a very high population growth rate. And over time with deer exclusion, we see that the population is declining, a significant decline. It's below 1, which is an exciting thing if you want to get rid of garlic mustard. And the density of garlic mustard goes down. This is a every 2-year graph because it's a biennial species. And so we score it every 2 years. So what is the practical information? Deer facilitate high population growth rates of garlic mustard. So if you have an infestation of garlic mustard that's just beginning, it's great to get rid of it. Pull it out if you can and stop it. And that garlic mustard and Asian stiltgrass are indicator species of high deer abundance. And I didn't put the slide in the right place, but I'll get to it. So now I'm going to move on to the effects of deer on unpalatable species. What we saw at Trillium Trail is that there was a change in the relative cover of garlic mustard and Flurkia, which is a very short-lived annual. And there was a decline in 3 of the unpalatable species that we were keeping track of in our paired plots. And Jack in the Pulpit is one of the ones that we considered kind of a star, and we decided to follow what was going on with that species. So is it true that unpalatable species, we thought unpalatable species should be experiencing success relative to the palatable species, or the unpalatable to deer. So again, we went to 6 sites up in northwestern Pennsylvania, and you can assess the activity of deer in an area by looking for the browse stems of Trillium, and they will never eat Jack in the Pulpit. It's full of oxalate crystals, and they feel bad when they eat it. So what we did was, in these 7 sites, we quantified the percent of the Trillium browse. So we had low browse sites all the way to very high browse sites on Trillium. And we used this as an indicator of the deer use or the deer impact in the area. And this is the thesis work of Chris Heckel. And one of the wonderful things about Jack in the Pulpit, among many others, is that it goes from vegetative, and it looks a lot like a Trillium when it's non-flowering, but then it shifts to be flowering. And the smaller individuals that are flowering are male first, and it's less expensive to produce pollen than it is to produce ovules and seeds. So the small flowers, once they're big enough to be reproductive, are male, and then they switch to being female once they grow bigger. So there's a size-dependent sex expression in Jack in the Pulpit. So in these 7 sites, we measured demographic characteristics. So reproductive plant biomass went down as the proportion of Trillium browsed went up. Seed rain, that is the amount of seed production per meter squared, went down as the proportion of Trillium browsed went up. And the sex ratio switched amazingly. It went from almost 50-50 at the low browse sites to no females in the highest browse site. And we were, again, as I said, we were very interested in understanding this mechanistically. So we did a number of measures of soil compaction, dryness, and some other things. And we found that when we combined these soil metrics into a principal components analysis, we found that the soil quality goes crashing down as the proportion of Trillium browsed goes up. So we were like, wow, this is really disturbing, because we were hoping that they would benefit from the being unpalatable. And so I contacted Bill McShay, and at the time he had his the exclosures and control plots up for 18 years. And I asked him if we could come over there and sample his plots, specifically looking at the species that deer don't eat. And what we found is that there was the plants that were in the deer, oops, this is supposed to say exclusion, deer exclusion plots were significantly larger, and the soil was significantly more compacted relative to the deer access plots. So we saw, not only did we see declines at Trillium Trail in the experimental plots, we saw them in northwestern Pennsylvania, and we saw them in Virginia. So it seems to be a general phenomenon of decline of these species. When we look at the stable stage distribution back to the 7 populations in Pennsylvania, what we see is, if we bin them into low deer impact, intermediate, or high deer impact sites, you see that this proportion in the stable stage distribution, when you project the populations out into the future, you see that the fraction of females is really low. This little orange sliver in the high deer impact sites. And that the transitions from the different, among the different stages should look like this, basically, in a low impact site. But in the high impact site, you see that the females are not staying females, they're reverting to males or vegetative. And mostly we see vegetative individuals. So it's kind of depressing. Lots of other studies since then have found similar abiotic effects of deer on the soil quality, on the light reaching the forest floor, on the mycorrhizae. And so this is a general phenomenon where having overabundant deer removing the vegetation, browsing, that it changes the dynamics of the soil community. So high deer abundance also suppresses population demography of unpalatable native plants, which is not what we expected to see. And this occurs through deer's indirect negative effects on the abiotic environment: compact, drier soils, more light to the forest floor, other things like this. So these are all practical things that can be measured, noted, or censused, even from citizen scientists, to keep tabs on what the deer are doing. And this is what was supposed to be earlier, that regionally, now we're talking about the forest understory community shifts, regionally, the deer are negatively affecting the forest understory communities. This was a compilation of 23 deer research sites that were across east, central, and northeastern the United States, so mostly forested habitats with a wide range of deer densities and invasive plant abundance and identity. And the results showed that the native and total plant richness, abundance cover, all the things that we think about, were lower in deer access versus deer exclusion. And that among the deer access plots, that the native species decreased. I mean, everything was more negative as deer density increased. And that indicator species analysis revealed that deer access favored some introduced plant species, including garlic mustard and microstegium. And you saw in Bairn's work and in my work that these deer are creating environments that favor the invaders. Okay. This sets up the question of whether there are differential and interacting impacts of invasive plants and white-tailed deer. And this is a review that Bairn and I both participated in. And it's a very fascinating read. I recommend it, but there's just so much clear evidence that they're tied together. I'm not saying that deer or, that invaders are more important than deer or less important than deer, but they're tied together in their effects. And one way that we know that we have a lot of deer around is the number of times that people get into car crashes. So these are data from the end of September from State Farm Insurance, which they keep data and post it every fall about car collisions. And where I work at Trillium Trail Reserve, we're number 5 for crashes, 1 in 62 chance of hitting a deer in Pennsylvania for a driver. West Virginia is number 1. Virginia is in the top 10. And all of these very dark colored states are high risk. So I think it's really interesting when you make a map, as I did, of here's the current microstegium distribution. So microstegium was introduced about here, and it's spreading. You can see that there's if you remember the previous slide, there's a lot of overlap with high density of deer and high density of microstegium. And the same is true for garlic mustard, for Allegaria, that there's this huge overlap in the states where deer have a lot of impact on forests and the abundance of garlic mustard. And I know this is like sort of a hand wavy thing, and I'm all about hard data, but I think that it's pretty clear that these things travel together. So, I want to show you a little bit about how deer facilitate Allegaria, and Allegaria produces antimicrobial chemicals, and how that can alter forest understory communities. So, most of the forest herbaceous species are highly dependent on mycorrhizal fungi. They partner primarily with arbuscular mycorrhizal fungi, and they are many of the showiest and most beautiful wildflowers that everyone loves to go and see in the spring make this partnership. And in fact, a lot of the species, this is the rhizome of false Solomon seal, and this is the rhizome of Trillium, and you can see that they lack root hairs, and the fungi, I think, function as root hairs for these species. So the way this mutualism works is that the carbon that is gained by the plant from photosynthesis goes to reproduction and storage and growth, but also gets shunted to the mutualus below ground. So all this white fuzz is supposed to be the hyphae of the mycorrhizal fungi. And what happens is that the plant is photosynthesizing away with light and CO2 uptake and shunting a substantial amount of its carbon to the fungi in its roots, the mutualists. In return, the fungi provide the plant with water and soil nutrients. And this is what the inside of a, if you look inside of a root, this is what you'll see if the plant has mycorrhizal fungi. So there are these arbuscules and vesicles. So this is arbuscular mycorrhizal fungi. That's how they're named. So they live inside the root, and the arbuscule, if you look up close, it looks like the alveoli of your lung. It's a site of exchange of nutrients and carbohydrates between the plant and the fungus. And this fungus extends its hyphae outside of the plant and draws up nutrients and water. And this is a very triangular view of how it works. So if there's a native plant, it's shunting carbon to the structures inside of the root, and also allowing that fungus to grow an external hyphal network. And the hyphae are sucking up the nutrient and water and giving it to the plant. So this is a typical resource benefit. Whereas if there's a plant that produces antifungal compounds or antimicrobial compounds like garlic mustard and many other species, what happens, the proposal was that it knocks out the external hyphal network, and the internal structures are still there creating an internally higher carbon cost because the native plant is shunting more carbon to these internal structures and more carbon to try to rebuild the hyphal network, which is being knocked out by the chemicals, and a decreased soil benefit, soil resource benefit for the native plant. And we've done a lot of field experiments, greenhouse experiments, all kinds of experiments, lab experiments, to show that this is really what is going on. We started doing this work about 10 years ago. And so the interesting thing was we were wondering if, in fact, this could result in a shift in the community at Trillium Trail. And one of my graduate students, Morgan Roche, used the long-term data that we have on species abundances. We do regular surveys of the different species at Trillium Trail. And she found that there was a significant increase in the non-mycorrhizal species at Trillium Trail. So it means that those species, the majority, which are in this mutualism, this nutritional mutualism, are paying a cost but are not getting the benefit, are declining in abundance, whereas then those that are not part of that mutualism are doing better. And it just seemed like, oh no, this can't be, could we really be measuring this? And so Morgan was able to use the Critical Trends Assessment Program data for sites that were part of the forest monitoring between 1998 and 2018. And in Illinois, what you're seeing is the red dots are places where there was at least, one record of garlic mustard being found, and the black dots were no records of garlic mustard. And what she found was that there was an increase in abundance in the non-mycorrhizal species across Illinois where garlic mustard is present. So it seems that the result that we've seen at Trillium Trail of decline in the mycorrhizal dependent species may be a general phenomenon. So, overabundant deer are a force of disturbance and change, which we all know, but they seem to be facilitating invaders that can change the forest understory community, affecting not only the herbaceous species that I've talked about here, but the woody species that I haven't talked about. And so I think that this is a very, we know this is going on, and that's why we're attending this symposium, but I'm hopeful that some of these results that we've shown you about the mechanisms and the details of why things are happening will give you some tools to measure things in the forest, in the field, that are good indicators of impacts of deer. And I'm happy to take some questions now. And I want to just add one more thing, and that is that one of the students that I worked with in a class that was like conveying science to the world, kind of as a communicator and an advertiser, came up with this graphic that I think is really good, that people don't really realize that we spend $750 million annually in the United States on property damages caused by deer, that, it goes on and on. 26 million lost to produce. Yeah, so we all know about this, but I think it's a really nice graphic and I wanted to share it.
Thank you
Okay.
for that, Susan. That's
Okay.
great. And thank you for a fantastic presentation. One quick, I think we have time, we have 1 more minute for maybe one question, and then we can allow you to answer those maybe behind the scenes, the rest of them. First one is, any sense how long individual plants can survive in the dormant demographic stage based on what you shared here? With the recovery of flowering plants you saw when you established exclosures, I would assume that the level could be substantial enough to initiate a recovery.
Yes, that can be very substantial. In fact, some of the individuals of fall Solomon seal would be dormant for 5 or 6 years and then pop back up. And they're very large plants, and they you know, the rhizome can fill your dining table, and you know, one rhizome would, part of the rhizome would send up a shoot, and then another part would send it up. So yeah, that can be substantial.
Wonderful. Great. Well, thank you so much. And there are a few questions here that you're, during the break or after break, you can answer at your leisure. Really appreciate it.